Good morning, everyone. I'm very pleased to introduce Dr. Raible today from University of Washington. Dr. Raible received his Bachelor of Science from Cornell University and his PhD from University of Pennsylvania. He joined the faculty at the University of Washington in 1995. He is currently a joint professor in Department of Neurobiology and Biophysics and professor in Department of Otolaryngology, Head and Neck Surgery. His lab investigates zebrafish as a model for hearing loss for investigating hair cell development, death, and regeneration. Again, welcome, Dr. Raible, and we're looking forward to your talk.
Well, thank you very much for inviting me. I'm pleased to tell you about the work that's going on in our group here at the University of Washington. I'd like to start off with something I think we all know, and that is that hearing loss is both prevalent and devastating. And these numbers from this recent study from the World Health Organization really bring it home, and that is that the global burden of hearing loss affects a rather amazing number of people, estimated at something like 1.5 billion people. That's across the board in terms of types of hearing loss, but even if you're looking at from moderate to severe on, it's still a very large number of people affected by this common and devastating disease. If you look at the distribution of cases, again from the WHO study, you see, however, that the vast majority of hearing loss is related, as shown over on the left-hand panel of this graph, to those who are growing older, that is aging presbycusis. And in fact, the vast majority, again, is related to this aging phenomena. And that's seen in the distribution of causes shown on the right-hand side of this graph. Those that, when affected early in life, are often affected by congenital genetic-related hearing loss. Those in the early years are also complicated by otitis, while those in later years is really related to age-related hearing loss. And in fact, the connection between hearing loss and other aging diseases has also become well known. For example, this data showing relationships between hearing loss and dementia. The study that was published in The Lancet just a couple of years ago showed that hearing loss actually can account for 8% of the vulnerability related to dementia. And in fact, the hazard ratio for dementia increases quite dramatically with increased hearing loss, as shown on the right from this study from Griffiths et al. So not only is hearing loss in itself isolating to people and elderly as they grow older, its association with other devastating conditions such as dementia make it a complex and vulnerable population that we're dealing with. In my lab, we're mainly interested in the fact, thinking about hearing loss is associated with environmental insult. I think we're all familiar with the idea that noise is a major cause of noise over exposure is a major cause of hearing loss. But exposure to environmental chemicals also, these environmental insults result in damage to the mechanosensors of the system, the hair cells. And in my lab in particular, we've been studying ototoxicity. That is loss of or damage to hair cells due to exposure to chemicals that would be otherwise useful therapeutic drugs. And the best known of these are the two classes, are the aminoglycoside antibiotics, and these include antibiotics such as neomycin, kanamycin, amikacin, and then the chemotherapeutics, the platinum-related chemotherapeutics such as cisplatin. Both of these classes of drugs are incredibly useful, and in fact can result in curing of the diseases that they're associated with, but the unwanted side effect results in irreversible hearing loss due to the damage of hair cells. So sometimes people ask, why study ototoxicity? There's a bunch of different reasons. One is, can we potentially come up with candidates for therapeutics? I think some of you are familiar with the work that's been done on sodium thiosulfate, for example, uh, which has been used to alleviate hearing loss due to chemotherapy with cisplatin. Uh, Pedmark is not now on the market for preventing this type of hearing loss. We're interested in the underlying vulnerability of the hair cells. If we can understand hair cell damage, for example, we may be able to come up with ways to alleviate that. But in fact, a third reason, more fundamental reason in some ways, that is trying to understand what is the underlying vulnerability of hair cells to these ototoxic chemicals as a way of understanding something about their function. That is, that what they normally do leaves them open to toxic exposure. And so it's potentially a way of also understanding some basic aspects of hair cell biology. As mentioned, my lab has for a long time used the zebrafish lateral line system as a model for hair cell biology. Here on this slide on the top, I show a picture of a zebrafish at about 5 days old. This fish is already freely swimming and feeding and interacting with its environment. It's about 3.5 millimeters long, so they're not very big. And you can see in this transgenic line that um, the hair cells of the lateral line system are labeled. Each one of these greenish dots is a cluster of hair cells on the surface of the body. So the aquatic vertebrates, such as zebrafish, have two types of hair cells. Fish have an inner ear, just like other vertebrates, that's involved in both hearing and balance. And in addition, they have the mechanosensors on the surface of the body that the fish uses to detect flow of water. This allows them to orient in a stream, a process known as rheotaxis. It allows them to detect movement in water, uh, in a sense, sometimes described as touch at a distance. The lateral line system allows the fish to be able to detect prey, for example, or advancing predator by these mechanosensors on the surface of the body. On the bottom here, I show a picture of one of the clusters of hair cells called a neuromast, where the hair cells are in the center, shown in magenta, and surrounded by supporting cells, glial-like cells that are important for their function in green. And if you kind of rotate the stack, I think you get an impression how the mechanosensory apparatus at the apical end of the hair cell sticking out into the water can be deflected by fluid movement, resulting in cell depolarization. And at their basal end, hair cells are innervated by sensory neurons in a fashion that's remarkably similar to those of the inner ear. And in fact, if you look at these hair cells in the electron micrograph shown here on the left, you can see that they have a characteristic bottle shape, apical mechanosensory apparatus, similar to those, again, of hair cells of the inner ear, actin-rich cuticular plate, shown here, basal situated nucleus, and specialized ribbon synapses that are important for high fidelity communication with the innervating sensory neurons. And in fact, we and others have identified mutations where these hair cells are not functional. And when the genes underlying these mutations have been identified, they've turned out to be zebrafish orthologs of human deafness genes, genes important for congenital hearing loss in people. And these include genes like the cadherins and that are important for the tip links, for example, that are important for the initial mechanotransduction, parts of the mechanotransduction channel like LHPL5 and TMIE, uh, genes that are involved in regulating sensitivity to mechanotransduction, like the Usher type genes, Clarin and Myo7, as well as parts of that ribbon synapse. So even though these cells are distributed quite differently than those in the inner ear, we think at a cellular level and a genetic level, they serve as a good proxy for uh, for hair cells in the inner ear, with the added advantage that because they're on the surface of the body, we have both access to manipulate and to visualize their function and dysfunction. And one of the simple experiments that we did was ask whether these hair cells are vulnerable to exposure to those ototoxic chemicals. And here, for example, showing the relationship between doses, increasing doses of aminoglycoside, neomycin, and uh, uh, consequent loss of hair cells. And in fact, we can see that this dose response relationship, rather simple, is actually um, turns out to be really quite powerful. And that is that, these error bars here are in fact standard deviations, and this shows that this relationship then is both robust and reproducible. And it gives us the opportunity to ask, under what conditions can we change this relationship? Can we find conditions that shift this dose response curve to the right, offering protection against hair cell damage, or shift this dose response curve to the left, revealing underlying susceptibilities. In addition, we can follow what happens with exposure to drugs. Here on the left, I'm showing a video where we're taking frames every 30 seconds. We've exposed hair cells that we're imaging here in vivo in the animal to the aminoglycoside antibiotic neomycin that's been tagged with a fluorophore that allows us to follow its entry into cells, first apically and then basally. And then we can see this catastrophic loss of hair cell integrity uh, that is the result of aminoglycoside exposure. One might notice that at this exposure level, which is at 50 micromolar neomycin, that you can see that some of these cells live and some of these cells die. And we're interested in why there are differences in the way hair cells respond uh, to these vulnerable compounds, potentially as a way of understanding how to alleviate toxicity. So as I said, my lab's been interested in this process of hair cell death. We've used the lateral line system to study it and a number of different aspects, seeing loss of hair cells very rapidly after exposure to aminoglycosides. We're also interested and have a large program to study hair cell regeneration, how it is that the zebrafish lateral line system can recover from this ototoxic exposure, this loss of hair cells in a really catastrophic manner, and yet can regenerate the cells so that there's functional recovery within 48 hours of exposure. I'm not going to spend a lot of time talking about this regeneration aspect, but just in our view, though, we kind of think of these as hand to hand, that there's something about the resulting damage that induces hair cell regeneration. And so, in some ways, both studies end up being complementary. Now, trying to understand why hair cells are susceptible to damage, a number of years ago, Robert Esterberg in the lab was interested in the potential vulnerable point within the cell, the compartment known as the mitochondria. And in fact, using, again, in vivo imaging, Robert and others in the lab were able to show that mitochondrial calcium accumulation after treatment with aminoglycoside antibiotics underlied the resulting loss of hair cells. So as we're seeing these again in this video here, we've targeted the genetically encoded calcium indicator GCAMP to the mitochondrial compartment itself in each one of these hair cells, exposed the animals to neomycin, and we can see this dramatic increase in uh, mitochondrial calcium that occurs before the loss of the hair cells themselves. Through a number of studies that I won't show now, Robert and his colleagues were able to show that the calcium that accumulates in the mitochondria was sourced from the endoplasmic reticulum, another intracellular store of calcium, that this excess calcium resulted in the production of reactive oxygen species, and blocking either the increase in mitochondrial calcium or blocking the increase in reactive oxygen species was sufficient to protect the hair cells against damage to neomycin exposure. And so, taken together, then these studies suggested that one of the underlying vulnerability points then of hair cells is their mitochondria. And to try and understand the relationship between mitochondria and hair cell function, Joy Sebi in the lab used physiological experiments to test the responses of mitochondria to normal stimulation. That is, a mechanical stimulus that we could give to the intact hair cells of the lateral line using a water jet. And I think you can make that out here. We've brought in an electrode, and with a pulse, you can see we can displace the mechanotransduction apparatus and study its response to a mechanical stimulus in vivo. Joy used both two different targeted calcium indicators to ask what were the changes in the calcium compartments in both the cytoplasm, shown on the left-hand side, and again, this mitochondrially targeted calcium indicator shown in the center panel. And we can see the responses of both the cytoplasmic and the mitochondrial accumulation of calcium in response to a water jet stimulus shown by this bar here. And we could see that these responses were coordinated but distinct. We saw that the increase in cytoplasmic calcium occurred rapidly and was tightly regulated in response to the water jet stimulus, both in onset and in recovery. By contrast, the mitochondrial calcium signal showed a delay in the initial increase in calcium, and then the mitochondrial accumulation of calcium again was delayed in its recovery. And so together, these experiments suggested that the mitochondrial calcium response was a normal response to, in fact, an underlying mechanical stimulus, and in fact, a mechanism in place to translate mitochondrial calcium signals into changes in mitochondrial energetics. That is, allowing a highly active cell like a hair cell responding to its function, mechanotransduction, by balancing its energy production was an underlying principle of hair cell function, and therefore the co-opting of this pathway by exposure to aminoglycosides co-opted this vulnerability, if you will, to result in hair cell death. So in a second set of studies, then we were interested in what is it about hair cells that make their mitochondrial network so complex and resulting in this underlying vulnerability. And so Andrea McQuaid, a former postdoc in the lab who now has her own lab at the University of New Mexico, used serial block face electron microscopy to reconstruct the mitochondrial network within the hair cells. And so using serial block EM, this allows us to do three-dimensional reconstruction of hair cells by using machine learning type approaches, which then can give us a very highly detailed view of the hair cells themselves, of the mitochondria within the hair cells, and how these mitochondrial networks are organized and how they respond to distinct conditions. Andrea was able to show that the mitochondria of the hair cell are indeed complex, and the networks that we see in the hair cells is characteristic of their function. When we compare a young, newly born set of hair cells to mature hair cells, we can see that there's an increase in the overall mitochondrial volume. And indeed, within individual hair cells, we see that there are, in fact, complex mitochondrial networks, individual mitochondria connected together, network that Andrea termed the max mito. These are shown in here in this panel in gold color, and you can see in the mature hair cell that these max mito networks are complex. They're situated ventrally, and they show this intricate association with the ventral synapses, the communication points between the hair cell and the innervating sensory neurons. As the hair cells mature, we can use stereocilia length as a proxy for hair cell maturity. We can see that the mitochondrial networks increase in size. We can see that the individual max mito, this basally located network, also increases. If you think of it in terms of z-score, that is uh, um, um, deviation from the mean by a standard deviation measurement. And you can see that, in fact, hair cells have a much more complex network than the surrounding support cells, either the centralized support cells that interdigitate between the hair cells or the peripheral support cells that act as the precursors of new hair cells. So again, this mitochondrial biogenic network is established during the development of the hair cells and increases in complexity as the hair cells mature. We were curious about the fact that we saw this increase in complexity associated at the time the hair cells became active in terms of mechanotransduction. And Andrea was curious about whether there was a link between that functional activation of hair cells, the onset of mechanotransduction, with changes in the mitochondrial network. And to explore this aspect, she took advantage of one of those mutants I was talking about, a mutation in CAD23, a component of the tip link complex within hair cells, and also a Usher's syndrome gene in humans. And what so the question was, what happens to the development of the mitochondrial network in the absence of mechanotransduction activity? Remember from the experiments that we did using the mitochondrial calcium indicators, there was a suggestion that mitochondria themselves are responding to active mechanotransduction. If we take that away here genetically, we wondered whether we'd see the increase in mitochondrial complexity that occurs during the maturation of the cells. And in fact, Andrea found two things. One, that in fact, the increase in overall mitochondrial volume, shown here in this left-hand graph, was unchanged in CAD23 mutants compared to wild type. That suggests that the process of generating new mitochondria, mitochondrial biogenesis, was actually unlinked to activity-dependent signals, which was rather a surprise. Although mitochondria have been heavily studied in terms of biogenesis in other systems, perhaps best in the cardiac system, there the link between mitochondrial growth and activity was direct, and here this suggests that the two are uncoupled. By contrast, the number of mitochondria, the increased complexity in the individual mitochondria, or the growth of this ventral mitochondrial network, the max mito, both are actually heavily impeded by the loss of mechanotransduction caused by taking away the tip link component that is necessary for the response of the hair cell to this external stimulus. And so, taken together, then these experiments suggested that there is a link indeed between the patterning of mitochondria due to mechanotransduction activity. And taken with Joy's experiments showing this increase in mitochondrial calcium, we're now interested in actually determine whether there's a direct interaction between calcium and mitochondrial patterning. And these are experiments that Andrea is now taking on in her own group at the University of New Mexico. So we're then at this point, I've given you a synopsis of experiments that we've done to show this vulnerability of the mitochondrial network underlying aminoglycoside toxicity, and that the mitochondrial network, the growth and patterning, and indeed responses to mechanotransduction are linked to its underlying function. We were particularly interested in the idea that maybe differences in the mitochondrial network were linked to this differences in vulnerability of hair cells to distinct damage. Remember, I said at some aminoglycoside exposure levels, some hair cells can live while others die. And in fact, we were turned to trying to understand whether there were, in fact, differences in mitochondrial metabolism that were linked to this distinct differences in vulnerability. So, in a series of studies done by Sarah Pickett in our group, we used another genetically encoded indicator of mitochondrial function, a protein known as mitotimer, to look at relative differences in mitochondrial metabolism. So here we're seeing the mitochondrial network in the hair cells of one of these neuromasts, this now familiar rosette of hair cells that you've seen in the figure so far. And you can see that this mitotimer, a protein that changes irreversibly from green fluorescence to red fluorescence upon oxidation, is indicating that different hair cells within the rosette show differences in oxidation levels. So some of the cells show green mitotimer signals, suggesting relatively low levels of oxidation, while others show higher levels of mitotimer signals, showing orange and red hues, indicative of increased mitochondrial oxidation. And in fact, there's a link between hair cell age and hair cell oxidation. Just as I showed earlier that young hair cells have a relatively simple mitochondrial network compared to mature hair cells. In fact, we also see this link between uh, relatively young hair cells showing greener mitochondrial signal, that is, less mitochondrial oxidation, compared to older cells that are red. That is, that there could be an accumulation of mitochondrial oxidation as hair cells age. Sarah was interested in the potential that there's differences in mitochondrial hair cell function and their hair cell susceptibility to damage. And in fact, we can see that in another one of these videos here where we're looking at which cells are going to survive an intermediate dose of aminoglycoside exposure. And as you can see here, I guess the arrows were, I should have said spoiler alert, that in fact the cells that show the highest levels of mitochondrial oxidation were the ones that were most vulnerable. And we can look at that in over multiple hair cells, over multiple groups, and you can see while there's relative differences between the two, if we look at the red to green ratio, we can see that those that live have a much more green appearance, that is, a lower red to green ratio compared to those that die. And in fact, you can measure the log odds, and in fact, the redder a hair cell is, there's an eightfold increase in susceptibility to hair cells exposure to aminoglycoside damage. So taken together, then, we think that we've learned something about the vulnerability of hair cells to aminoglycoside exposure, that the mitochondria are a vulnerable point within the hair cell to aminoglycoside damage, and that this is potentially due to the fact that the mitochondria are responding to hair cell activity as part of their normal function. Now, we'd love to be able to develop a drug that might leverage these mitochondrial studies to prevent hair cell damage to occur. But in fact, interfering with mitochondrial function is probably not the best way to go about protecting cells, given the necessity of this compartment for their normal function. So we took a completely different approach, and that is to just screen for compounds that potentially could protect. And these experiments were done by Felipe Santos and Henry O, research fellows in the lab, and also Anna Vlasits, a post-bac scholar who's now gone on to graduate school and her own scientific career. And here the idea is that the zebrafish themselves, being small, 3.5 millimeters long, we can actually put them in 96-well plates and score whether compound libraries could prevent the hair cells from damage to aminoglycosides. So in each one of these wells, then we have a zebrafish, and in most cases, aminoglycoside exposure causes the hair cells to be killed. And when we looked for wells where, in fact, we saw that even in the presence of aminoglycoside, the hair cells were prevented from damage. And indeed, this screen resulted in the identification of a compound that we initially called Proto-1. This is a small molecule that is from one of these chemical libraries that we could show that increasing concentration of proto, shown here on the x-axis, results in survival of hair cells on the y-axis, kind of the opposite dose response graph I showed you earlier when looking at the aminoglycoside exposure. And again, being able to look at large numbers of animals and the robustness of the assay, here we're showing two different runs of the same assay showing the overlap, meant that we could think about doing a structure activity relationship, making changes in the underlying chemical itself, and ask whether this changes the dose response relationship here now for protection against aminoglycoside damage. And this iterative chemical approach using zebrafish to screen for new possibilities resulted in the generation of this compound shown down on the bottom right, a compound that was a new chemical entity, had not been made before, and resulted in the compound that resulted in the founding of our small company, Auricula, that I had labeled as my disclosure at the beginning. And along with Ed Rubel's group, then we showed that this compound could be translated from zebrafish to a rat model. Here we're looking at hearing loss in terms of decibels in response to rats being treated with the clinically relevant aminoglycoside amikacin. And you can see that in this black line here, amikacin exposure results in hearing loss across the stimulus frequency spectrum, low frequency to high frequency, from 20 to 50 decibels worth of damage. When we treat with the small molecule at the same time, we could show that there's dramatic and significant protection across the frequency spectrum. And so these encouraging results made us think that perhaps this was a molecule that could be translated to the clinic. We were interested in trying to understand what the function is. The advantage of doing a phenotypic screen is, you know, in the end that you have a compound that results in protection, in this case against aminoglycoside damage, but you don't necessarily know why it's doing that. And indeed, we still to this day don't have a clear understanding of what aspect of hair cells this small molecule is interacting with. However, we know that the underlying mechanism by which the compound protects hair cells is by changing how aminoglycosides actually end up getting into cells. So here's another couple of movies that show, again, labeled aminoglycoside on the left, entering into hair cells and resulting in the damage, just like we saw earlier. And in a set of hair cells being exposed to the aminoglycoside, you can see over the same time frame and same doses that in fact the small molecule 13661 prevents the entry of aminoglycosides into hair cells. And this then suggests that it's affecting this early step in hair cell dysfunction. That is that the aminoglycoside needs to get into the hair cells to cause the hair cells to die. And this suggests that the compound is interfering with hair cell damage by this unique feature of hair cells themselves. That, in fact, one of the reasons why hair cells may be so susceptible to damage is that they differentially accumulate these aminoglycoside drugs compared to other hair cells. Sorry, compared to other cell types, so that the hair cell becomes differentially vulnerable. Although you can't see it, these movies show hair cells in situ, that is localized in an animal. The surrounding black includes other cells, but those cells are not normally taking up aminoglycoside. And in fact, one of the reasons why aminoglycosides actually are effective antibiotics is that their highly charged nature prevents them from entering normal cells usually, and therefore allows them to be efficacious in interacting with the bacteria, the infections that they are being used to treat. We went on to show this effect of 13661 on aminoglycoside uptake in zebrafish was also true in mammals. Joe Bellairs, again, a resident who did a year of research in the lab, showed that if we turn now to looking at aminoglycoside accumulation in the mammalian inner ear, that we could see that the small molecule prevented the accumulation in inner ear hair cells. So on the left-hand panel, we see a kind of classic view of the cochlea, where we see a row of inner hair cells at the top, 3 rows of outer hair cells. And if we expose to the aminoglycoside G418 tag with Texas red, you can see that there's accumulation within hair cells of the inner ear. And in fact, the outer hair cells are differentially accumulating the drug. This was interesting, of course, because it is the outer hair cells that are the most susceptible to aminoglycoside damage in both animal models and in human patients. And co-treatment with increasing doses of 13661 results in a reduced accumulation of the toxic aminoglycoside in hair cells, shown here over on the right-hand graph, so that you can see that there's increased doses. So again, taken together, these experiments suggest that the mechanism of action, although we don't still understand the specific molecular interaction involved, uh, the mechanistic interaction is the block of differential uptake of aminoglycosides into hair cells. So taken together, these studies were encouraging, and we have now moved on to uh, completing preclinical and clinical research so that we can move towards a phase 2 trial. As I showed, we have good efficacy. The 13661 protected against all the aminoglycosides we tested in zebrafish. So we tested 10 different aminoglycosides. We showed in vivo efficacy in a rat model, repeated doses of amikacin exposure. And I didn't mention it, but parenthetically, 13661 does not alter the antibacterial effects of aminoglycosides. It does not protect bacteria from aminoglycoside exposure. That would make it a rather useless way of thinking about co-treatment. We moved on to do safety and toxicity studies, both using a rat CNS safety model, a dog cardiovascular safety model, and in both rat and dogs, we did 28 and 90-day GLP toxicology studies. All of those were completed. They were all necessary for FDA to um, allow us to classify 13661 as an investigative new drug, an IND, and move then, because it was a new chemical entity, to a phase 1 trial, which we have now completed. In the phase 1 trial, this was with healthy adults. We did 2 different studies, a single ascending dose study, that is increasing doses of the 13661 compound. And this allowed us to understand the pharmacokinetics of this compound within healthy human patients. And we found it was actually quite different than what we had seen in the rat, for example, but allowed us to understand both the Cmax and the AUC were both dose proportional to increasing doses. We saw increasing accumulation. And this allowed us to then think about what would be a good therapeutic scheme. We did a multiple ascending dose study as well. So we adjusted the initial daily dose regimen. Because we see this long accumulation, we could dose at 50 mgs, and then with a follow-up dose of 10 mgs per kg. And so we could show that, again, we could develop steady state concentration within these healthy subjects after a day. We only saw a small amount of vulnerability for safety, and that is at the very highest doses. We saw the potential minor prolongation of QT, so a potential for heart arrhythmia. We didn't see effects like that in the dog, and these were minor and only at highest doses, although it's something we will be concerned about monitoring as we move forward, of course. But we found that a 10 milligram dose is predicted to be efficacious compared to the rat study, and allowing us to have a good therapeutic index. So what is the phase 2 trial going to be? It's whether we could see changes in caused hearing deficits for people who are being treated with aminoglycoside antibiotics because they have infections from non-tubercular mycobacteria. This pulmonary disease, NTM, is actually due to inhalation of mycobacteria from environmental sources. Sometimes it's called hot tub lung. In England, it's often referred to as Lady Windermere's disease because of the persistent cough, I guess, due to the play by Oscar Wilde. So NTM, the mycobacteria are environmentally sourced. And although people who get the NTM have a persistent infection, uh, in contrast to tuberculosis, these NTM are not transmissibly spread from person to person. Rather, it's an environmentally accumulated source. And in fact, there are multiple NTM species of bacteria, and they can be treated with multiple different regimens of antibiotics, but in the end, some are resistant to most antibiotics, and then aminoglycosides is then part of the standard of care. Patients, and about 30% of patients who have NTM end up being treated with aminoglycosides. And when they're treated for a long time. These are treatment times of 60 to 90 days. And as you might expect, then more than 70% of these patients treated with aminoglycosides showing hearing loss. Like TB, NTM results in these granulomas that are found in the lung, these kind of walled off induced macrophage structures that prevents the bacteria from ready access by the host. And one of the reasons why these persistent infections are so pernicious. Uh, from a patient population standpoint, the fact that more than 70% of the patients treated with these aminoglycosides are showing moderate to severe hearing loss means that we actually have a pretty good signal to noise ratio. That is, we should be able to see whether co-treatment with our compound is actually going to show some protection. That is because the change from baseline is so high. Moreover, there's no standard of care. You know, the patients are just treated with aminoglycoside. There's nothing to prevent. So even a small but statistically significant protection against hearing loss will be of added value. So the trial is just underway. When I say just underway, it's taken quite a few steps to get to that point. We have 5 different study sites. It's mainly being run out of Oregon Health Science University, but National Jewish Health in Denver, the Mayo Clinic, uh, Medical University of South Carolina, and UT Tyler are sites again that where this trial will be taken up. It's a trial that's funded by NIDCD, and the collaborating institutes include NIDCD. Uh, the drug manufacturing was done through an agreement with the National Center for Advancing NCATS, National Center for Advancing Transitional Science, and sites are all now approved. The drug is manufactured in capsule and complete and has been now delivered to the sites. And I was hoping I'd be able to say today that we've enrolled our first patient, but already 2 of the sites are fully approved to have patients, and we're expecting first patient this month. This is pretty exciting for us. This is kind of the endpoint of a long set of studies that started with our zebrafish studies, has moved to the clinic and potentially uh, um, helping these vulnerable populations who are being exposed to aminoglycosides, cured of their disease, but with the underlying challenges of hearing loss. We think that results from these long-term exposures can also be expand potentially the use of aminoglycosides for more short-term exposures. There's clear evidence that short-term exposure of aminoglycosides also results in hearing loss, but the variation makes it quite challenging to study. So we think results in these studies will allow us then to open up towards that. So with that, I'd like to thank you all for listening. The group here that has made the work happen. Again, the work on 13661 result in the formation of a small company, Auricula, for which I'm a co-founder, that allowed us to move this from a zebrafish lab into a clinical trial.